This is a production of Cornell University.
I'm very excited to be here. I'm also a little uneasy because, as Kevin mentioned, I am very broadly trained macroecologist. Sometimes I think I'm a conservation biologist, definitely not a plant scientist. So bear with me. Some information may be spotty in terms of maybe what is the core interest for you. So to put my presentation in perspective, it's all about new ways to study or investigate an old question. Why are species distributed or found where they are? And I'd like to start with an inspiring quote that pushes biologists, or at least in 1985, E.O. Wilson was pushing biologists to take on this grand challenge of trying to describe and trying to learn more about diversity in general worldwide. So in the words of E.O. Wilson, this is a mission worth all our effort. So that was 1985, not too long ago. Then in 2010, another great entomologist came back to tell us that it is critical, it is very important to try to study diversity on our planet, and maybe what we need is a grand approach, the type of approach and the type of funding and resources that NASA has. So, some years ago, 15 years after E.O. Wilson called all scientists to study diversity and focus on that, 15 years later, the message was very similar or even stronger. Now, another message that is related to trying to understand, trying to study diversity and basically provide a justification for this effort is from the world of ecosystem science and valuation of ecosystem services. So the idea here is we do want to know as much as we can about species, and it is worth investing our time because our understanding of species distribution and function and ecosystems actually in the end overall affect human life quality. So basically put a spin to our type of research that makes the public more interested and more aware of why we study diversity patterns. Okay, just to give you a very brief schematic view of what I do, and then a cloud of words that are more common or abundant in my publication so far. I am, as I said, broadly speaking, interested in species distributions and factors that affect species distributions. And I use several ways of addressing questions that are related to species distributions. And then our understanding of species distributions does branch out into many different fields, and I'm just naming a few here: conservation biology, biogeography, and disease ecology, the more recent ones. Okay, so what I want to do today is give you an idea of the kind of work that I do and maybe spark some interest in your using some of these methods for your own research. And my plan is to go down a spatial scale from broad scale questions to maybe regional all the way to fine scale. You'll notice that I have one here, the finest scale question in gray area, gray font, and that is because we are just starting to work more on this at this level, on this scale, and we have more questions and more data to collect and analyze than actually giving you answers. Okay, now in order to address questions at these various scales, questions related to distribution and factors that affect species distributions, I'm using two main types of research techniques. One is ecological niche modeling, and the other one is remote sensing. So I'll give you very brief overviews on the two as we go down the scale to see these examples. These are plant examples, as I said. They may not be very dense and informative from a biological standpoint, but hopefully you can help me fill in the blanks in those areas. Okay, so ecological niche modeling, or E&M, is a very active field of research. But what I wanted to do first is start with just an understanding of the terminology, because we know that niche can mean various things to various people. So you may be aware of over the history of ecology, you may be aware of definitions of niche provided by Grinnell in the early 1900s, Elton in the 1930s, and then Hutchinson in the 1950s and 60s. So how do we make sense of all these definitions? To me it makes sense, and to other people it makes sense to think of the effects that organisms have on environments, on the environments that they occupy, versus the responses that organisms have to the environments and the kinds of requirements that organisms have, how they choose environments in which they live and survive. Okay, so if we think about the niche from this effects versus responses, or impacts of species on the environments versus requirements, environmental requirements of species, maybe it's a little easier to make sense of these definitions. Okay, and then trying to put the niche concepts on a geographic scale, we can think of questions that are addressed at small scale extents, geographic extents, versus large geographic extents. And on this sliding scale, going back to impacts versus requirements, think about at the small geographic extent, when we talk about species impacts, what we are talking about is the functional role of species at that small scale. And for these kinds of questions, we usually use mechanistic models that try to quantify the dynamic relationships between species. Think of consumer resource dynamics. So those are very different models than what I do, which is at larger extents, and ecological niche modeling is more useful at these larger extents. I should point over here, where the interest is in the requirements that species have in order to survive in a certain region. So that summarizes the geographic niche, or the abiotic niche of a species. And to do that, we use empirical models, and I'll show you one type of models that we use in this broad umbrella of ecological niche modeling. And the big assumption here is that the requirements, the environmental requirements of the species, are stronger than the interactions, the biotic interactions. So basically, the abiotic interactions are more important than biotic interactions in shaping the distribution of species at these larger scales. Okay, so how do we go about, if we are into the ecological niche modeling field, how do we go about measuring, estimating, quantifying this abiotic or Grinnellian niche, as it is known in the literature? Well, there are three methods that I'm aware of, or three possible approaches. One would be physiological experiments. So we go in the lab and we test the limits that a species has, an organism has, to, let's say, varying temperature, if it's an aquatic species, varying salinity, so on and so forth. So we go into the lab and we find out what are the physiological boundaries of the species, which can then possibly be mapped on geography. This is really hard work to do, and if we are interested in mapping the biodiversity of a little study, of a study that has not been, of a country that has not been studied very well, we do not have time to develop physiological models for many different species for which we don't have information. Then Slightly maybe mad scientist scenario would be to introduce species in various types of environments and figure out which kinds of environments allow that species to survive in long term. This is obviously impractical at large scale, probably unethical. We don't want to change ecosystems by doing these kinds of large scale studies introducing species. So the third method that is widely used in ecological niche modeling world has to do with making use of information we have available about where species are and the environmental conditions that are present at those locations. So we build correlation type models, simply put, that quantify that relationship between where species are and what kind of environments are present at those locations. And just to give you more visual on these correlation approaches, to estimate environmental requirements, we start with species records. These could be museum specimens. These could be surveys, any type of occurrence data. Then we, on top of that, we use various digitized maps of environmental conditions, and I'm showing you just some examples here. Precipitation, annual precipitation, annual temperature, seasonality of precipitation and temperature, so on and so forth. And then these form our calibration data or training data, which we use to build a model, estimate the niche of that species. And there is a plethora of algorithms and approaches to making sense, quantifying this relationship between organism presence and environment. I'm naming just a few here, and I'll briefly mention show Maxent. Okay, oops, sorry, jumped one. So once we have that, once we have the model calibrated with these occurrences and environmental data, what we do is we then project this model back onto geography. So we take data out of the geographic context, the spatial context. In this environmental context, we build a model, we calibrate the model, and then we take that model back into geography, but we project that model. And I'll get back to why this is important, this model projection. But what we get is an estimation of suitability across the landscape for that particular species of interest. And I need to move a lot faster. Okay, so one of the widely used algorithms is Maxent. The paper that promoted the use of Maxent or described this algorithm was published in 2006. It has been cited over 3,000 times now. It is a widely used methodology. What we do here is basically contrast known presences with the rest of the environment. So we have some occurrences, we have some knowledge of where species is present on a landscape, and we contrast the conditions at presences with the conditions at across the rest of the landscape. And that's one way of quantifying suitability. We get a probability of suitability more likely if the conditions are similar to presences, and less likely if the conditions are similar to the rest of the background. So it's contrasting presence and the rest of the environment. Okay, so jumping to the first example of the use of ecological niche modeling, I put this at the largest scale, but you'll see that we actually varied the scale of the question for this project. And the idea was, can we create reliable species richness estimates when we are dealing with various geographic extents, or the scale of our analysis varies. And this is an area of intense research, because we use these estimated species richness maps to create, for example, hotspots for conservation or for future climate effects, so on and so forth. So knowing how well we estimate species richness with ecological niche modeling is of importance for various reasons. Okay, so the question is, do we get various estimates in terms of quality, various species richness estimates from ecological niche modeling when the scale varies? So that was the question that we tried to answer. AJ Harris is not a PhD candidate anymore. She successfully defended about 2 weeks ago, so she's moving on to a postdoc at the Smithsonian. But she was the main driver of this particular study, and then her advisor, Dr. Mike Palmer, both from the botany department at Oklahoma State. And we are working, the manuscript is in review. Hopefully we'll have positive criticism and useful reviews back soon. Okay, so what was the idea here? The idea was to correlate observed species richness with estimated species richness. So the observed species richness, these data come from floras, and the estimated species richness, the data come from stacked models. So we build basically, sorry, I'll talk about the models later. But okay, so what is the observed species richness? This is what we consider the standard in this study. So using a large amount of data from various floras that, a lot of data came from the Floras of North America project, but also from checklists and very small scale floras across North America. We, AJ mostly, AJ amassed a data set of Cactaceae and Pinaceae species in North America, north of Mexico. So excluding Mexico, we didn't have data for Mexico. So what you see are at the county level, but the floras had different scales, but at the county level, you see the species richness, the number of species in these two families that we decided to compare. Okay, and then based on the information that we had about the floras, we created 6 classes of extents, or floras that were generated at 6 different extents, starting from 10 hectare, between 10 hectare and 100 hectares, all the way to 1 million to 10 million hectares. And we try to have a very well-balanced data set. We split the flora information in the 3 regions, Arctic, Atlantic, and Pacific. And we had in each scale class, we had the same number of floras that we considered for this study. Okay, and then for the estimated species richness, we applied ecological niche modeling. Species by species, we build models for the Cactaceae and Pinaceae with the same occurrence data, environmental data approach, Maxent ecological niche modeling algorithm, and we created these predicted distributions for each species, which then we stacked for each flora. Okay, so what did we get? Oh, sorry, and I forgot to mention we had 24 species of Cactaceae and 51 species of Pinaceae. Most of the data for the ecological niche modeling part of this project came from the Global Biodiversity Information Facility, which is a distributed database of museum records and observation. And then we had, as environmental data or variables, we had climatic data and soil data for this project. Okay, so we generate these stacked models to estimate the number of species in each flora we considered at these 6 different scales. So then we put the 2 together and we try to see how well they correlate. Okay, starting with Cactaceae, what we see is that if we pool all extents, those 6 or 8 different extents we have, if we pull them together, we get a good correlation, I would say, 0.68. Deviation from the slope of 1 quite big, so it is okay. It's an okay correlation, not a very strong correlation. So correlation between observed species richness through flora and estimated species richness through this stacking of ecological niche models. But when we start separating by extents of floras, what we see is at the largest extent, that 1 to 10 million hectares, we have a high correlation. So at the largest extent, this seems to be a useful approach. Now, on the other end of the spectrum, at the finest extent, abysmal. Let's not do this. Let's not use ecological niche modeling and the stacked approach to estimate species richness because it's not really doing good. With Pinacea, the situation is not good at all. So at all extents, definitely not working. At the largest extent, somewhat working. The correlation is 0.56, somewhat okay, but not a great approach. The slope, the deviation from the slope is better. When we go to the smallest extent, as you expected, do not do this. Let's not do this. Let's not estimate species richness with the stacked ecological niche modeling approach. Okay, so that was one way of informing this ecological niche modeling field of the applicability of this approach to estimating species richness. Next, what I want to do is go into a slightly smaller scale and talk about climatic niche dissimilarity for one plant species that has two morphs. So the question here was, do we see a climatic difference between populations that have different morphologies? And this study is a collaboration with botanists, again, botanists from Janet Steeds from OSU, then Diana and Naoki from University of Alaska Fairbanks, and then geologists from OSU. So what we did here is, not me, but Janet, examined two morphs of Arabidopsis kamchatka, and she used the herbarium specimens available at the University of Alaska. So the two morphs that she looked at and that are common in Kamchatka populations are the trichome-bearing leaves versus glabrous leaves. So smooth versus leaves that have these unicellular trichomes. So she was able to find 116 specimens for the trichome-bearing morph and 230 specimens for the glabrous morph. And this is a map of the distribution of these specimens. She georeferenced, she and Jeff georeferenced many of these occurrences. Okay, so what did we do with this? We took the records for the two morphs, environmental data again, and we built two different models for the trichome-bearing specimens based on the trichome-bearing specimens and the glabrous specimens. So what we see is, first look at these maps, is the trichome-bearing has a narrower distribution than the glabrous.
Coastal, maybe?
I'm sorry?
Coastal, maybe?
Yes, exactly. This is more coastal, and this is more inland species, a wider distribution based on the occurrences and also based on our estimated suitability for the glabrous. Okay, but then so we have a visual of the climatic suitability for the two morphs, but we want to quantify how different, are they really different, these two suitability envelopes. So what we did is we used background similarity tests, which allow us to tell if the two morphs are ecologically more similar or less similar than expected by chance. And to do that, we created 100 replicates of these models, where we randomized one data set for one morph and created models, and then randomized the other data set for the other model, the other morph, and created models. So basically we have real models based on real data, comparing that with random models based on randomizing, taking at random taking at random locations that are not presences for that particular morph. And then we use two statistics, Schoner's and I statistic. What they basically do is they look at the probability of suitability for one morph, and then the probability of suitability for the other morph at each location in geography. And they do slightly different things, but overall they should be they should provide a unified answer. If we get different answers from these two different metrics, it means that we are not doing something right. So the idea here is quantifying the suitability probability for each pixel and comparing between the two morphs. So what do we get? What we get is that this is the distribution of random models, and this is where our observed overlap, niche overlap or similarity between the two morphs is. So this is number of replicates. Obviously, we have just one for the real models, and then we have a number of 100 each for the random models. So this is telling us that there is a statistical significant distinction. The two morphs are less similar than expected by chance based on the statistic. We did the same for this other statistic, and we obtained similar results where the niche dissimilarity between the two morphs was statistically significant compared to a null distribution of these values. Okay, so what we are saying is the two morphs are climatically dissimilar. The one other way to look at this is which variables contribute most to the two models, the two real models for the morphs. And this is a list of the variables that we used. And what we see is that in bold, we have variables that cumulatively amount to 70% or more contribution to the model gain of that for that particular morph, meaning that the variables in bold have the highest contribution, the most important contribution to a good model fit for that morph. So what we see is that there is a discrepancy between glabrous morph, which has in that highest contribution mean diurnal temperature range and temperature seasonality as very important, but not the trichome bearing. And then going down here, we have annual precipitation and precipitation seasonality for the other morph, the trichome bearing morph. So this tells us that one morph is more sensitive, at least from this modeling perspective, one morph is more sensitive to temperature variation and the other one to precipitation. And elevation and minimum temperature of the coldest month was in common between the two morphs. Okay. Next, I would like to talk a little bit about discrimination of trees and then habitat structure. So going down this scale, moving to finer and finer scales. To get to these questions, now we are switching to approaches that are based on remote sensing, a very, very big field in engineering and geography. And we are making use of it just a tiny bit. There's a lot of potential to develop this field. Just to get everyone on board, we have some sort of sensor, can be a satellite or can be on an airplane. And then what the sensor is recording is the amount of light that is reflected off different surfaces. That's the basic of remote sensing. Why is this important? Well, for us, it's important because it allows us to get data from above that we don't get on the ground. It is accessible data. If it's satellite, collected by a satellite, and it's free like Landsat, we have access to that data. We have a greater temporal resolution because the satellite keeps orbiting the Earth, so we have a really good temporal resolution. And we do have this potential in extracting biodiversity data that is of use to us. And just to give you some ideas, we detect type of surface with remote sensing data. So based on the amount of light reflected, we have different types of, two different types of vegetation, and then man-made, let's think of man-made surfaces. So the amount of light that is reflected is basically what gets used in these land cover classification if you look at the land cover map. Also very important is the amount of light that is absorbed or reflected by different pigments in the structure of the plant. And this is where remote sensing becomes extremely important for us biologists who are interested in plant function, plant structure, plant distribution. So this is the important aspect of remote sensing. Now, one other important aspect of remote sensing is that there is a wide variety of sensors collecting this information, this amount of light that is reflected off various surfaces. So a multispectral sensor splits the wavelength, the electromagnetic spectrum, in a few distinct bands. So think of Landsat, for example. We have a few distinct bands. The information, the sensor can only process information, amount of light reflected from this one particular band, for example, here. So that's multispectral. Now, hyperspectral refers to splitting this electromagnetic spectrum in very narrow bands, 10 nanometer bands, so we get hundreds of bands. These sensors provide a data-rich data sources that are rich in information. Okay. So one way of using this is to try to look at particular reflectance of different species, and in this case, trees. So here the question is, can we pick out emergent trees, big, large canopy trees that are standing above smaller trees? Can we pick them out spectrally from one of these hyperspectral, high density of reflectance bands? This work was done in collaboration with excellent botanists from Peru and a field assistant whose name I forgot. I'm really sorry. This was a long time ago when I was finishing my PhD. So these are botanists in the field. Then Greg Asner is a scientist at Carnegie Institution for Science. And since I have my botanist collaborators in field mode, pictures, I didn't have them in more official pictures. I decided to go with field mode for everyone else. So Greg Asner, then my former PhD advisor, and my collaborator from World Wildlife Fund. And this is, like I said, work that was finished as I was finishing graduate school. So this has been published some time ago. The study site was somewhere in southeastern Peru, beautiful area. Also highly diverse area in terms of tree species. And for me, the interest was a very small subset of these tree species that are used by various animal species as food resource. So frugivorous animals that need resources, fruit produced by these trees, and actually the seasonality, which I will briefly mention. So these are the species of interest. We have Papeş species here, and I forgot the family for the Brazilian nut. I apologize, but we have 4 taxa from Papeş and then the Brazilian nut. And then a map of showing location, various points for these species. One thing to notice is low sample sizes. These are hard areas to work because they are so diverse, the forest, and you can walk and walk and walk and not find a second emergent Apuleia tree, for example. So the sample sizes are less than ideal. We also had satellite images, these hyperspectral, high band density images from 4 different times. And you notice that precipitation-wise, we had dry season, more rain, and then the wettest part of the season, of the year, and then another from the dry. So we had both dry, more dry, and then one image from the wet season, which was quite impressive, and I was quite happy to get that one because of the cloud cover. It's really hard to get images for these regions. Getting the hyperspectral imagery is exciting, but then the processing is really convoluted. I won't go into it. I'll just say that from that great 242 band data set, we were down to 126 that we used because of various reasons: cloud, atmospheric problems, poor signal to noise ratio. So quality of information made us reduce that initial data set by half of almost. And then we did various things to it. I'm not going to go into it, but just wanted to mention this 126 bands. And just a view of how this Hyperion hyperspectral imagery maps. So we have a crown here that is almost an entire pixel, 30 by 30 meters. Another one that is almost 30 by 30 meters. And then we have a tree here that is not a full pixel. It is split between 4 different pixels. So we were focusing on these pixels that represented more than 40% of the crown of a single tree. And then this is a spectral profile with the bands removed, the remaining 126 narrow bands that we kept in the analysis. Okay, so we did a linear discriminant function analysis of these 126 bands. We initially did a stepwise selection procedure, so we used only the most informative of these 126. And we got really good classification errors, meaning really low classification errors. So what you see is for one tree species that it's actually we couldn't identify to the species level, so we used the genus. We have, for this one taxon, we have classification errors, meaning that the linear discriminant function analysis was not able to spectrally identify the individuals that well. But we also had only 4 individuals, so it's pushing the limits of this analysis. And then for the Brazilian tree nut, we had, in one of the time frames, remember we had 4 different images, we had error. Okay, and just to show you the 2 profiles for the various individuals, but we do see differences in these spectral curves. We also see temporal variation within a taxon. So here's Apulea in July and in December, different spectra. And that difference was quantified with a canonical analysis. So what we see is that based on the season that these images are collected, the spectra group in various parts of the canonical space. What this means is we don't have a spectral signature that is species-specific at all times, and we can use that spectral signature to identify a tree. It is season-based. Okay, lastly, the habitat structure. This is going to be very fast because I don't have a lot of information. We've just started to work on this. And this is with my PhD student, Cassandra, who took on this challenge of learning how to use LiDAR, terrestrial LiDAR, to quantify vegetation architecture for one particular species, bird species that she's interested in. So this is the bird species. This is a general view of the shrubland grassland ecotone in Oklahoma. And this is what she's trying to quantify. The LiDAR, or light detection and ranging, what it does is it sends pulses out and then measures the time it takes for the pulse to reach the sensor again. So based on that, if we think of a laser or LiDAR that goes, or laser beam that goes from above, this particular structure will have this form where there is more returns when it hits a part of the tree and less returns when it goes through between 2 trees. Now, we can do this from above, but we can do also this from the ground. And this is where our interest lies for now. How can we do this terrestrial scanning of bushes and quantify vegetation structure? And these are some of the variables that we hope to take out of these clouds of points that we get: mean shrub height, shrub volume, foliage profile. Yes. On this is distance from the scanner. This is how much time it takes for the beam to get back to the pulse to get back to the sensor. So it's time. The time is converted to distance. Okay, so we don't have any answers. This is what we've been doing this summer, collecting data. Very difficult logistics we can talk about later if you are interested. But what we are doing is we are scanning one way and then scanning another way to get a full 360 view of the vegetation from this point all the way 500 meters distance. And what we get are, for now, pretty pictures. This is a point cloud where vegetation is classified, so we get rid of soil or any other atmospherics, anything that is not vegetation. And this is a profile of one of the study sites. This is another pretty image. So far, no quantitative information, but this shows vegetation height classified. So the lightest green is the highest vegetation, and the darkest, or the brown pixels are the shortest vegetation. So we can get vegetation height for now from these images. Okay, so what I tried to do was show you an example at large scale of ecological niche modeling of estimating species richness. And what we see is that, at least for the two families we studied, fine scale do not do this. Large scale, Cactaceae works better than Pinaceae. Then moving on to a more regional scale, we compare the morphology of one species, two different morphs, and the potential adaptation to different environments. And what we showed was that we have two morphs that are statistically, they are climatically dissimilar. And then here we were able to identify trees trees based on spectra, but there is seasonal variation, so we cannot use one spectral profile to just go there and map species. It has to be season specific. And then habitat, we don't know yet. We are still in the process of gathering data and making sense of it. But in general, what I want to close with is say that these types of approaches, ecological niche modeling, for example, allows us to basically extend the amount of information we can gather from occurrence data and environmental data. So it allows us to learn a bit more about ecological requirements, climatic requirements for particular species. So we hope that this improves our understanding of species distributions, why species are where they are. And then at fine scale with these remote sensing approaches, what we are trying to get at is more of the species function locally and interactions in more of a habitat, plant-animal interaction type. Any questions?
This has been a production of Cornell University, on the web at cornell.edu.